Hello everyone, welcome to a new session of the paper Cell Biology. In this session, we would study about cellular locomotion, role of cilia and flagella in movement. We would also study about the process of cell death and cell senescence. The process of cell differentiation in plants and animals would also be studied. Cell locomotion. Amoeboid movement. It is the most common mode of locomotion in eukaryotic cells. It is a crawling type of movement accomplished by the protrusion of cytoplasm of the cell involving the formation of pseudopodia. The cytoplasm slides and forms a pseudopodium in front to move the cell forward. This type of movement has been linked to changes in action potential. The exact mechanism is still unknown. This type of movement is observed in amoeboids, slime molds and some protozoans such as Naegleria gruberi, as well as some cells in humans such as leukocytes. Sarcomas or cancers arising from connective tissue cells are particularly adept at amoeboid movement, thus leading to their high rate of metastasis. Flagellar movement. A flagellum is a lash-like appendage that protrudes from the cell body of certain prokaryotic and eukaryotic cells. The word flagellum in Latin means whip. The primary role of the flagellum is locomotion, but it also often has function as a sensory organelle, being sensitive to chemicals and temperatures outside the cell. Flagella are organelles defined by function rather than structure. There are large differences between different types of flagella. The prokaryotic and eukaryotic flagella differ greatly in protein composition, structure and mechanism of propulsion. However, both are used for swimming. An example of a flagellate bacterium is the ulcer-causing Helicobacter pylori, which uses multiple flagella to propel itself through the mucous lining to reach the stomach epithelium. An example of a eukaryotic flagellate cell is the mammalian sperm cell, which uses its flagellum to propel itself through the female reproductive tract. Eukaryotic flagella are structurally identical to eukaryotic cilia, although distinctions are sometimes made according to function and or length. Three types of flagella have so far been distinguished: bacterial, archaeal and eukaryotic. The main differences among these three types are summarized as follows. Bacterial flagella are helical filaments, each with a rotary motor at its base, which can turn clockwise or counterclockwise. They provide two of several kinds of bacterial motility. Archaeal flagella are superficially similar to bacterial flagella, but are different in many details and considered non-homologous. Eukaryotic flagella, those of animal, plant and protist cells, are complex cellular projections that lash back and forth. Eukaryotic flagella are classed along with eukaryotic motile cilia as undulipodia to emphasize their distinctive wavy appendage role in cellular function or motility. Primary cilia are immotile and are not undulipodia. They have a structurally different 9+0 axoneme rather than the 9+2 axoneme found in both flagella and motile cilia undulipodia. Flagellar arrangement schemes. Different species of bacteria have different numbers and arrangements of flagella. Monotrichous bacteria have a single flagellum, for example, Vibrio cholerae. Lophotrichous bacteria have multiple flagella located at the same spot on the bacteria's surfaces, which act in concert to drive the bacteria in a single direction. In many cases, the bases of multiple flagella are surrounded by a specialized region of the cell membrane, the so-called polar organelle. Amphitrachus bacteria have a single flagellum on each of the two opposite ends, that is, only one flagellum operates at a time, allowing the bacteria to reverse course rapidly by switching which flagellum is active. Peritrichous bacteria have flagella projecting in all directions, for example, E. coli. Ciliary movement. A cilium is an organelle found in eukaryotic cells. Cilia are slender protuberances that project from the much larger cell body. There are two types of cilia, motile cilia and non-motile, or primary cilia, which typically serve as sensory organelles. In eukaryotes, motile cilia and flagella together make up a group of organelles known as undulipodia. Eukaryotic cilia are structurally identical to eukaryotic flagella, although distinctions are sometimes made according to function and or length. Motile cilia. Larger eukaryotes such as mammals have motile cilia as well. Motile cilia are usually present on a cell surface in large numbers and beat in coordinated waves. In humans, for example, motile cilia are found in the lining of the trachea or windpipe, where they sweep mucus and dirt out of the lungs. In female mammals, the beating of cilia in the fallopian tubes moves the ovum from the ovary to the uterus. Ciliates are microscopic organisms that possess motile cilia exclusively and use them for either locomotion or to simply move liquid over their surface. Primary or immotile cilia. In humans, primary cilia are found on nearly every cell in the body. In comparison to motile cilia, non-motile or primary cilia usually occur one per cell. Nearly all mammalian cells have a single non-motile primary cilium. In addition, examples of specialized primary cilia can be found in human sensory organs such as the eye and the nose. The outer segment of the rod photoreceptor cell in the human eye is connected to its cell body with a specialized non-motile cilium. The dendritic knob of the olfactory neuron, where the odorant receptors are located, also contains non-motile cilia, which are about 10 cilia per dendritic knob. Muscular movement is the method used in most of the vertebrates, including man. Muscular movement is based on the use of muscle fibers. Muscle fibers have the unique property of ability to contract and relax, which exerts a force. This force is responsible for the movement of body parts and locomotion. Muscles constitute nearly 40 to 50% of the total human body weight. Muscles have the unique properties of excitability, contractibility, extensibility and elasticity. In man, muscles are classified into three categories: skeletal muscles, smooth muscles, cardiac muscles. Structure of skeletal muscles. When a typical skeletal muscle is viewed under a microscope, it consists of many elongated cylindrical cells called muscle cells or muscle fibers. Each muscle fiber is enveloped by a plasma membrane called the sarcolemma. The sarcolemma surrounds a quantity of cytoplasm called sarcoplasm. Within the sarcoplasm are many nuclei and a number of mitochondria. Skeletal muscle fibers are thus multinucleate. Also, within the muscle fiber is the sarcoplasmic reticulum. The mitochondria are called sarcosomes. A highly magnified view of skeletal muscle fiber reveals thread-like structures which are about 1 to 2 millimeter in diameter called myofibrils. These bear the characteristic cross striations. The myofibrils are stacked in compartments called sarcomeres. Narrow zones of dense material called Z-lines separate sarcomeres from one another. A dark anisotropic band, also called A band, is present in the center of the sarcomere. Adjacent to this lies a light isotropic band, also called I band. Alternate arrangement of dark and light bands produces the striated appearance to the skeletal muscle. At the center of the A band, a less dark zone called H zone is present. In the center of H zone, M line is present, made up of fine threads that connect the myofilaments. The Z line is present at the center of the I band. The sarcoplasm has many thick and thin filaments. In each sarcomere, the thin filaments are present at the two ends and the thick one at the center. The H zone contains thick myofilaments only. The I band is composed of thin myofilaments only. The rest of the A band has both thick and thin filaments. The thin myofilaments are composed mostly of the protein actin. The thick myofilaments are composed mostly of the protein myosin. Both are contractile proteins. Actin has low molecular weight and it occurs as G-actin and F-actin. The thin filaments also contain two other protein molecules, tropomyosin and troponin. The myosin molecule is shaped like a rod with a round head. Myosin rods form the long axis of the thick myofilaments and the heads form projections called cross bridges which connect to the active site present on the actin. Cell senescence and cell death. Cells continually experience stress and damage from exogenous and endogenous sources, and their responses range from complete recovery to cell death. Proliferating cells can initiate an additional response by adopting a state of permanent cell cycle arrest that is termed cellular senescence. Understanding the causes and consequences of cellular senescence has provided novel insights into how cells react to stress, especially genotoxic stress, and how this cellular response can affect complex organismal processes such as the development of cancer and aging. Cellular senescence is the phenomenon by which normal diploid cells cease to divide. In the cell culture, fibroblasts can reach a maximum of 50 cell divisions before becoming senescent. This phenomenon is known as replicative senescence or the Hayflick limit in honor of Dr. Leonard Hayflick, co-author with Paul Moorhead of the first paper describing it in 1961. Replicative senescence is the result of telomere shortening that ultimately triggers a DNA damage response. Cells can also be induced to senesce via DNA damage in response to elevated reactive oxygen species, activation of oncogenes, and cell-cell fusion independent of telomere length. As such, cellular senescence represents a change in cell state rather than a cell becoming aged, as the name confusingly suggests. Although senescent cells can no longer replicate, they remain metabolically active and commonly adopt an immunogenic phenotype consisting of a pro-inflammatory secretome, the upregulation of immune ligands, a pro-survival response, promiscuous gene expression or PGE, and stain positive for senescence associated beta galactosidase activity. The nucleus of senescent cells is characterized by senescence associated heterochromatin foci or SAHF, and DNA segments with chromatin alterations reinforcing senescence, also known as DNA scars. Senescent cells are known to play important physiological functions in tumor suppression, wound healing, and possibly embryonic or placental development, and paradoxically play a pathological role in age-related diseases. The elimination of senescent cells using a transgenic mouse model led to greater resistance against aging associated diseases, suggesting that cellular senescence is a major driving force of aging and its associated diseases. Programmed cell death or PCD. Programmed cell death is a normal physiological form of cell death that plays a key role both in the maintenance of adult tissues and in embryonic development. In adults, PCD is responsible for balancing cell proliferation and maintaining constant cell numbers in tissues undergoing cell turnover. In addition, programmed cell death provides a defense mechanism by which damaged and potentially dangerous cells can be eliminated for the good of the organism as a whole. Virus infected cells frequently undergo programmed cell death, thereby preventing the production of new virus particles and limiting the spread of the virus through the host organism. Apoptosis and autophagy are both forms of programmed cell death, but necrosis is a non-physiological process that occurs as a result of infection or injury. Necrosis is the death of a cell caused by external factors such as trauma or infection and occurs in several different forms. Recently, a form of programmed necrosis called necroptosis has been recognized as an alternate form of programmed cell death. It is hypothesized that necroptosis can serve as a cell death backup to apoptosis when the apoptosis signaling is blocked by endogenous or exogenous factors such as viruses or mutations. The concept of programmed cell death was used by Locomotion and Williams in 1964 in relation to insect tissue development around 8 years before apoptosis was coined. Since then, PCD has become the more general of these two terms. The first insight into the mechanism came from studying BCL2, the product of a putative oncogene activated by chromosome translocations of 10 found in follicular lymphoma. Unlike other cancer genes, which promote cancer by stimulating cell proliferation, BCL2 promoted cancer by stopping lymphoma cells from being able to kill themselves. Apoptosis. Apoptosis is the process of programmed cell death that may occur in multicellular organisms. Biochemical events led to characteristic cell changes, morphology and death. These changes include blebbing, cell shrinkage, nuclear fragmentation, chromatin condensation and chromosomal DNA fragmentation. Autophagy. Macroautophagy, often referred to as autophagy, is a catabolic process that results in the autophagosomic lysosomal degradation of bulk cytoplasmic contents, abnormal protein aggregates and excess or damaged organelles. Autophagy is generally activated by conditions of nutrient deprivation, but has also been associated with physiological as well as pathological processes such as development, differentiation, neurodegenerative diseases, stress, infection and cancer. Cell differentiation in plants. Differentiation in plants refers to the process by which distinct cell types arise from precursor cells and become different from each other. Plants have about a dozen basic cell types that are required for everyday functioning and survival. Additional cell types are required for sexual reproduction. While the basic diversity of plant cell types is low compared to animals, these cells are strikingly different. For example, some cells such as parenchyma cells retain the potential to respond to environmental and/or hormonal signals throughout their life, and under the right conditions can be transformed into another cell type, which is also known as transdifferentiation. Other cells such as the water conducting vessel elements undergo cell death as a part of their differentiation pathway and thus can never transdifferentiate to another cell type. Plant tissue systems. Each of the three tissue systems is found in a predictable location and consists of one or more multicellular tissues that carry out a unique function. For instance, the dermal tissue system is found at the surface of the plant and consists of a single tissue type, the epidermis. The epidermis functions to protect the plant from water loss, to permit gas exchange, and to provide a barrier to the invasion of harmful fungi and other microorganisms. The ground tissue system occurs internally to dermal tissue and may consist of three tissue types: parenchyma tissue, collenchyma tissue and sclerenchyma tissue. Cell differentiation and development. Cell differentiation is only part of the larger picture of plant development. As plant organs develop, the process of organogenesis, the precursors of the tissue systems form in response to positional signals. Then, within each tissue system precursor, cell types must be specified in the proper spatial pattern. For instance, the spacing of trichomes and stomats within the protoderm must be specified before their precursor cells begin differentiation. Exchange of signals among neighboring cells is an important aspect of the process of spatial patterning and cell differentiation. In addition, long distance signals are required so that the strands of xylem and phloem cells within the leaf vascular bundles connect perfectly with those in the stem. Cell differentiation in animals. In cell differentiation, a generic cell evolves into a specific cell type, which actually is a response triggered by the cells themselves. This process allows the single celled zygote to evolve into multicellular adult organism constituting hundreds of various types of cells. Cell differentiation has been found to be very critical in embryonic development and also plays a pivotal role in the functions of complex organisms all through their lives. Cell differentiation process changes the size, shape, metabolic activity, membrane potential and signal responsiveness of a cell. The changes occur because the gene expressions possess highly controlled medications. Apart from few exceptions, changes in the DNA sequence are never initiated by the cellular differentiation process. Due to this, the distinct physical characteristics in each cell is possible even though the genome is similar. During the developmental stage of embryo, cell differentiation allows the growing organism to produce array of cells in huge number for further need. When the organisms mature, they possess adult stem cells, somatic cells and germ cells. Due to this, the process of cell differentiation has become very important in relevance to the adult embryonic stem cells and for other general cellular process. Embryonic differentiation is the process of development during which embryonic cells specialize and diverse tissue structures arise. Animals are made up of many different cell types, each with specific functions in the body. However, during early embryonic development, The embryo does not yet possess these varied cells. This is where embryonic differentiation comes into play. The differentiation of cells during embryogenesis is the key to cell, tissue, organ and organism identity. Once an egg is fertilized by a sperm, a zygote is formed. The zygote divides into multiple cells in a process known as cleavage, triggering the beginning of embryonic differentiation. During cleavage, the zygote divides but maintains its size in the process. The zygotic division produces blastomeres which later make up the hollow sphere known as the blastula. Cells migrate within the blastula to locations that will later define the structure of the embryo and consequent organism. In this process called gastrulation, three germ layers arise: the endoderm, mesoderm and ectoderm. Cells in these three layers will give rise to different parts of the organism. The endoderm eventually becomes the gut. The mesoderm develops into muscle, the skeletal system, some organs and connective tissue. The ectoderm differentiates into the nervous system and skin. As the embryo continues to develop, individual cells continue to differentiate. These differentiated cell types are made from what were initially the same types of pluripotent embryonic stem cells. An assortment of physiological mechanisms guides certain cells towards particular developmental pathways, creating varying cell types. This is made possible by the cell's inherent ability to control what genes are expressed and translated into proteins. Every cell contains DNA within the nucleus, containing the blueprint to build many different proteins in the cell. Different signals can cause embryonic cells to select specific parts of the DNA, which can then be used to synthesize proteins, eventually building different cell types. Differentiation of cells in the embryo is brought about by both internal cellular factors as well as extracellular factors that act on the cell from the outside. Much remains to be understood about the exact molecular interactions that govern cellular differentiation. There are two main types of cellular development that pertain to embryos: mosaic development or regulative development. In mosaic development, which is not characteristic of mammals but of organisms such as annelids, differentiation occurs in steps that are set in order and progression without input occurring between neighboring cells. On the other hand, regulative development involves the interaction of adjacent cells within what is known as embryonic fields. The advantage of regulative development is the flexibility that it confers to differentiation. For example, a cell's pathway may change depending on the cellular environment in which it is placed, not merely by its internal mechanisms. The process of embryonic differentiation is crucial to proper animal development. The process involved in embryonic differentiation continue to be explored and have relevance to studies involving embryonic stem cells and in vitro cell differentiation. Let us now summarize what we have learnt in this session. Migrating cells undergo a series of characteristic changes in shape: extension of a lamellopodium or pseudopodium, addition of the extended leading edge to the substratum, forward flow or streaming of the cytosol, and retraction of the cell body. Cell locomotion probably occurs through a common mechanism involving actin polymerization and myosin 1 generated movement at the leading edge, assembly of adhesion structures, and cortical contraction mediated by myosin 2. Cellular senescence, a state of irreversible growth arrest, can be triggered by multiple mechanisms including telomere shortening, the epigenetic depression and DNA damage. Together, these mechanisms limit excessive or aberrant cellular proliferation, and so the state of senescence protects against the development of cancer. Recent evidence suggests that cellular senescence also may be involved in aging. Here are a few assignment questions for you to work out. 1. Elaborate on cell senescence and programmed cell death. 2. Explain cell differentiation in plants. 3. Which are the different types of flagella and explain their function. 4. Explain the structure of skeletal muscle. 5. Differentiate between apoptosis and autophagy. Here I can suggest a few books for your reference. The Cell, a Molecular Approach, written by Jeffrey M. Cooper, Robert E. Hausman, 3rd edition, published by ASM Press. Biology, written by Peter H. Raven, George B. Johnson, 5th edition, published by McGraw-Hill. Cell Biology, written by Gerald Karp, 7th edition, published by John Wiley and Sons. Thank you for watching this program. Let's meet in the next session with another topic. Till then, bye.